All right. Hello, everybody. Thanks a lot for coming. It's a real pleasure to have this workshop. Finally, with Michael, we have been interacting for a year now, and this is the first time I see his face online, and we almost finished a paper without meeting each other, so which is amazing, but it's good to do that at some point. All right, so this workshop will introduce, we have done workshops before in many circumstances and places about the tidy adapters for transcriptomics and genomics. And in this workshop, we would like to introduce the community we are trying to build, as well as how all this tidy infrastructure for bioconductor can interact on the genomic side and transcriptomic side. And a lot of development has been going on for cytometry and proteomics and so on.
Oh yeah. Before we start, quick logistics. The CAB TAB is moved back to what, 12:30?
12:15.
So yeah. So there's an update for when that's starting. It's not going to start right after this.
All right. I would like to show you a few things before we start with the actual hands-on part. So, a few announcements. We are trying to build this tidyomics community. So all people that like to work with tidyverse and want to apply this to their biological analysis. And part of this effort is to write a paper about that, which we have done and we are completing. And we want to invite, one thing that we thought to do is to invite the community to contribute to some of the outstanding challenges for this ecosystem, as well as propose some if you want, and participate in this way to the manuscript, which is in preparation and should be ready very soon. So the idea is to, this is a QR code that will go to the GitHub project with the open challenges. So feel free to take a picture to go to the link. And this will take you to this page, which is the actual challenges, and is structured in a GitHub project. So the idea is that these are some of the challenges we have realized, and these include the infrastructure, transcriptomics, genomics, and whatnot. And you can assign decide if you want to contribute to some of those and assign yourself to some of these. And many of these can be contributed by more than one person, so we can create teams and so on. And also you can feel free to, propose other features that you would like to work on, and just be creative. We are very open to that. And the contributors will be very welcome to participate again to this manuscript that we can share, and we will make public very soon. I think that's about this. Is pretty much it.
Oh, I guess one more thing is there's a Slack channel on. There's a channel on the Bioconductor community Slack where if you have any questions about this or about using this ecosystem, there's many people who are interested to, help out and give comments.
Very true. All righty. So we have the workshop is on the Bioconductor workshop platform, and of course is also on GitHub. So if you you can find it at github.com/tidybiology. Here we go. And you can find the name of the workshop is Tidyomics Workshop BioC 2023. And of course, you can download the package, install it locally if you want, and so on in a later time. And obviously, there is the render workshop if you want to follow that way, which is this. But we have opened an instance on the workshop platform, Galaxy. It's called, I'm not sure why the name was assigned to tidySingleCellExperiment, which is part of the workshop, but not all. But nonetheless, this is the name of the workshop. And you hopefully will launch soon. The workshop is very light.
Can you delete this?
Ah, that's true. Here we go. Okay, so maybe let me ask who is familiar with tidyverse and use it day by day? Okay, virtually everybody. Who has ever worked with any of the tidy adapters in bioconductor, transcript, tidySummarizedExperiment or the plyranges or others? Okay, good. So for everybody, you are in the right place. So we will do some introduction to these tools, but as well, Michael will go to how to integrate genomic and transcriptomic analysis through these tools. So, we cover all levels of interest, and the material here will be more details than what probably we will go through. So the material is also there to go for you at the second time and learn a bit more. All right, let me go here. Here is a bit description of us. This some of what some of the goals of this workshop, and also this workshop is also limited to in the scope. So it's very clear that we assume here we are somehow familiar with the tidyverse, and as well, this workshop doesn't want to be a guide how to do analysis. It's just a showcase of how you can use this tool to do analysis. So there is a case study, but that's the function. We have a couple of links. We decided to, in the interest of time, to not go through, but there is a good introduction to tidytranscriptome ecosystem and tidygenomic ecosystem. That anyway, we are doing what we can to put them together as a unique interconnected ecosystem. Good. Any question about something is not working or some technical things? Good. All right. So can I stand a bit? Okay. Hopefully everybody can hear. All right. So I will introduce. This is focused on single cell data, at least this initial part. But I want to show you, if you go to the tidy biology, is no repository, but organization, there is also a manifesto. There is a blog. And one of the contributions could be to make a blog post, by the way. There is a manifesto for tidytranscriptomics. So I just want to mention that although this will be limited to single cell, just briefly describe what the whole transcriptomic ecosystem is. So we have adapters from some of the popular containers in bioconductor, which is SummarizedExperiment, which is called tidySummarizedExperiment, and you will understand well what this is, and also for SingleCellExperiment, which is called tidySingleCellExperiment. Similarly, we have adapters for Surat. So basically, you can use tidyverse across technology. So to switch from bulk to single cell with the same grammar and style. Yeah.
Like I had problems before switching between the different versions of Surat and the SingleCellExperiment and back to specific analysis, like the time with Monaco. You guys have a way to get past that?
At some level, in the sense that these two tidy adapters do not do analysis. So you do analysis the way you did, but the data manipulation, yes. In many cases, virtually all cases, you can convert the object, run the same code in terms of manipulation, merging data set and so on, and it will run. In this case, yes, you don't need to, if you don't want, you can use the same grammar for all of it. But we want to stress that this is not a replacement for the, let's call it standard grammar. You can switch between one or the other as your convenience. And also tidybulk is for bulk RNA sequencing analysis, is a full, very comprehensive analysis framework that follow tidy principles and includes most of the tools and facilitate, cross-tool usage and so on. Okay, that was the premise of this. So let's go to coding. Where are we? Here. All right, so here we load some of the libraries that you might know, SingleCellExperiment, some tidyverse library and so on for visualization. And let's try to load the single cell experiment object we have in our material. And as you can see, you have the familiar representation of single cell experiment. So it's a summary where you get the dimension of your data set, some examples of features, samples, and metadata. Now, let's call the library tidySingleCellExperiment, and we print on screen the same object. So the object is untouched, but the representation is changing. So you have a flat table representation that includes cell metadata and reduced dimensions and so on. And this is a representation, but as I will show you, you can also interact with it as if it were a flat table, and the underlying object will be updated up to that. So you can, filter, mutate, and visualize based on principal components or metadata seamlessly. So in a way, we generalize the idea of variables. So everything here is a variable.
Can I have one comment at this point? Because Stefano built all of this infrastructure, and I'm like learning it. So one thing I realized, as he says, it's an abstraction. This has not unspooled the single cell experiment object. This is just a way to conceptualize the object so that you can operate on it with tidy. So the underlying object is there. Like, it hasn't changed the S4 object, if you know bioconductor. It's just a way to now operate it on, like, with an API.
Yeah, so no, there is no package that will ever know that these tidyomics will be loaded. There is no trace in the. So if you're yep.
Is it just like by importing or loading tidySingleCellExperiment? Is there just a new show function?
Correct.
Correct.
Exactly. And so the show function merges also the manipulation you do. So you have this consistent appearance interface. Yeah.
So this is the call data of the single cell experiment?
Plus reduced dimensions and plus relevant informations, for example, for spatial transcriptomics, there will be spatial coordinates and so on. So all cell relevant information, and I will show you. You can you can decide to add gene information if you want to visualize, calculate, and so on. All right, so that's the representation. If you want to use tidyverse verbs, but you are very, you like very much the original representation, you can toggle that. There is no problem. But of course, for the sake of the workshop and how we like it, we convert it back to a flat representation.
I have another question. So, have you, as part of this, have you implemented the multiplier verbs for the S4 data frame class in bioconductor?
I mean, it's not generalized at that level,
Okay.
but we implement this for the. I mean, what we've done is implement it for the some of the major containers, and we will keep doing that. I mean, this interesting question. I've never thought about generalizing that. Maybe it's possible, but S4 objects are, for G ranges and SummarizedExperiment are very heterogeneous. So
Yeah.
we make them somehow consistent through this interface. I'm not sure if possible to.
Yeah, I've looked into it before. It's non-trivial.
Yeah, I would imagine so, but maybe there is a magic bullet. I don't know. But it's a good question. It's worth to think about that. Okay, so just to again emphasize what Michael has said, all your code keeps working on this object because obviously it has not changed. So if you call assays, it will work. If you call whatever package the interface with SingleCellExperiment, it will work. No package will ever know that you have loaded a SingleCellExperiment library. Okay, so any question about this? I will show you some of the example of data manipulation. All right. So here I show you some very basic manipulation that you can do in tidyverse, apply to this object, but you can imagine that the power of this is connecting all this for doing very efficient workflow for manipulation, visualization, and so on. Let's say we want to filter some of the cells that are in phase G1. We apply the plyr verb filter. So I can run this, and you can see here that well, we state the number of features, cells, and assays. Here you can see the number of cells has changed from 3,000 to mostly half of it. What we can do as well, obviously, this is material for you to read. We won't necessarily read through it at any time. You can select some of the columns. For example, if you want just to visualize, display some of the columns, or you want to make the object actually lighter. In this case, we are not assigning to any variable, so it's just a temporary operation. And so we are selecting cell, file column, and some other column on the cell. You can see here the result. And this has updated the metadata, so has just preserved some of the columns. But we are not saving this in any way, so it's just for display in our case.
Question. So can you pipe that into as table just to get this view as a table?
Yes, it will be converted to table. Yeah, can do it now.
Assuming all the columns are compatible with a regular data frame.
Well, yes, I mean, also nested columns are preserved as the table allows.
Like, can you like put a geranges in a column of a bioconjure data frame? I don't know how.
Yeah, you can. We do it in SummarizedExperiment. We have tested on it, but I mean, it might improve. You can have definitely a nested column for geranges. In some cases, there are more than one range, so it's nested. In some cases, there is just one range, so actually you see it explicit in the table, and you can operate on it. I mean,
yeah, we do that later.
Yeah, we will
show a nested example later.
Yeah. There are some columns, such as reduced dimension, which are view only. So it does not make much sense of edit this reduced dimension. So these are there at any time, are just view only. You can select them out. Another thing we can do in this case is to polish our table or do concatenate operation. In this case, we are converting phase to lowercase. And creating new column, phase L. You can see here, we are just showing these three columns for convenience now. And the metadata has been updated accordingly.
Why the UMaps there? Like if you selected things that are...
Because those columns are view only, so they will be there. In the future, we will make this visually a bit more explicit, but basically, UMaps are grabbed from the object displayed. So since are view only, you cannot select them out because you would drop them from the object in this case.
So if I had lots of other reduced dimensions, would they pop on there?
Yes. Yeah. By default, we cut, for example, PC up to 5, but you can display them all. And is not that inconvenient as you can have a very big table and it will be, the columns that are not printed here, the names are printed downstairs. So it's in practice, it's a good solution. I found at least.
So will it cut everything to 5 by default?
By default, yes. The display, yeah, but you can toggle to print everything. One little example, still simple, but sometimes, for example, you have the file path you downloaded the data with, and there is some information here, let's say the sample ID. Okay, so with using, in this case, you can see I'm selecting the file path here. Using tidyverse is quite easy to do quite powerful operation. As well, I'm doing one by one, but of course you can concatenate all this once you have done, so it's quite clean and readable. TidyR offers this extract function, so you can use a regular expression to extract one or many pieces of information from this path. In this case, we are isolating the sample ID. This will create a column called sample. And here I'm showing to you this sample column that has been created from the path. Yeah.
Is there a place where all of the implemented tidyverse are documented so we know what we can use?
A place. Well, the package is all documented, but the documentation mirrors, for most part, the tidyverse one. The application is the same.
It's just plyranges.
So again?
Are these just coming from plyranges?
No. This what, sorry?
Like select mutate.
From dplyr.
Oh, okay.
Yeah, so that these are all functions that are included in the dplyr, tidyr, ggplot, and plotly.
Right. So are these like S4 methods for those functions, essentially?
No. Those are S3 methods we apply to.
You would also have limitations with mutate right there. Like you can't, for example, it will yell at you if you don't have the right amount of rows or whatever it is right there.
Everything that's, yeah, so everything that would make sense for the flat representation has the same behavior as a table. If that's what you mean.
Yeah.
Yeah, yeah.
These are, the views, the abstractions you're getting are tables right there on the underlying objects.
Exactly. So the behavior is mirrored completely. We abstract all these.
You're using virtual abstractions.
Yeah.
You can do stuff like arrange and reorder, and it'll reorder the whole summarized experiment or the.
Well, to be specific, single cell experiment in this case, yes. Summarized experiments we don't allow because we integrate transcript and sample is complicated. Yeah? But in this case, yes, there is no issue with doing that. You will reorder the cells based on whatever you want. And again, many of these could be transitive. You just do stuff to observe or display. Not all the time you arrange, you want to actually save that order. You can do many things on the spot. So one good thing about this way to do things that you don't have to have temporary variables if you don't want to. So you save a variable for a reason. You want to, it's big variable. You want to save it or reuse it. But a lot of operations do not require temporary variables. So it's quite elegant in this way as well. All right. Okay. So another just other examples of very quite powerful convenient function from tidyr is unite. Let's say you want to create unique IDs for doing pseudo bulk or any reason between sample ID, patient ID, cell type ID, whatever you want. You can use unite, same thing as you would do with a table. And as you can see, we are creating a sample ID, uniting these two columns, and here is your sample ID. Similarly, you can do separate, and you can imagine all the operation you do on a table. Good. I mean, how this was we have 3 parts. This was the first one. Any question on this first part before I do a little case study?
Could we use the character in the Slack? I mean, could we use character replace the variable in the Slack?
Yes, whatever tidySelect accepts, which is a lot. In this, the new dplyr likes any of, and you can use character. It complains if you just use character, but yeah, the answer is yes. Yeah, whatever dplyr accepts. Are we on time? So 24. I will go to this quick. Again, we don't have much.
Yeah, I'm not going to show all part 3.
Yeah,
Anyway, it's more than.
Yeah. So this was basically the beginner part, just to make you understand, and probably the most important. We have a case study that please go back and look. It's quite interesting. I'm not going to focus on everything, but basically this case study involves, after analysis, we have been asked to study better T gamma delta cells. And so this is something we did using tidySingleCell to create signatures, visualize signatures, filter data, and so on.
Remove it.
Oh yeah, don't use that URL, please.
All right. So long story short, a publication of Pizzolato proposes a signature for gamma delta, which is not well studied in breast cancer, which is what this data is. So we can use join feature to add this data to the representation, and that will be added to the metadata. And so now we can do arithmetics on this. We can visualize this, filter on this, do whatever we want. So again, we are generalizing the idea of variables. Everything here is a variable. You can integrate it the way you want.
So that's adding the expression of those genes in each cell as additional columns?
Correct. You can do also a long representation will output a table if you want to do faceted plot or whatever. But these represent
gene and then expression.
Exactly, but that's not anymore a single cell experiment.
Yeah.
Then you can use, will do whatever you want. But in this case, it's compatible with single cell, so it's just a single cell experiment. They propose an arithmetic, and so we quite elegantly we can do quite complex arithmetic using the mutate. In this case, the signature is the scaled sum of the positive marker minus the scaled sum of the negative. And we are creating just a signature column that I'm visualizing here. And now we can use common visualization functions from Skater in this case. So we can switch, of course, between whatever infrastructure we want. And we are plotting signature score. You can see that this blob of cells might be our gamma delta that we might want to isolate and analyze further. We do with this infrastructure is quite convenient if you want custom plots or more customizability for some reason to use ggplot directly on single cell. So that's what we are doing here, replicating the same plot. These are custom visualization, but you can see it's very straightforward here. You can see in this case we are arranging on signature to pop up the cells that have higher score. Choosing our color palette, that's it, basically the same thing. So choosing between ggplot and more standard ad hoc visualization, it's not a cost. It's very easy to choose one or the other. We do a quite bad thing, but for the sake of the workshop, we select these gamma delta cells selecting on a fixed score. Not a good idea, but it's quite simple to visualize here. There is also tidygate package, which allow you in a reproducible way and procedural to draw gates and create columns in that way. Anyway, no time to explain here, but we can talk later. And this is the alternative with base R. So you create a couple of temporary variables, you merge these temporary variables, and it's quite typical you can see here that you require quite a few commands. It's a bit less elegant, arguably so, than the tidy representation. This is just an example of how you can reanalyze. Again, you pipe your object into standard bioconductor functionality, and you integrate data back with the tidyverse. So you switch between one or the other with no problem. So just a single cell experiment will be returned at any point. So it's quite easy to do so. In this case, we are fast M&M is stripping some annotation. We are adding it back, running your map, and so on. So you have filtered and reanalyzed gamma delta cells quite in a straightforward way. I would say.
So just now in that code chunk, you said like left join as table as the object gamma delta. So do you have to convert that to a table?
In this case, I just want the metadata back. So you cannot left join a single cell experiment to another single cell experiment. You know what I mean? So I take the original single cell experiment, which has all the metadata. I just convert it to a straight table and left join to my single cell experiment, which has been stripped from the metadata. So you obtain the same.
But can you I think you can. There's a merge. There's a merge method for S4 data frames, right?
If you mean here, we are not merging cells. We are just merging
data frames.
Just the metadata,
Yeah, S4 data frames,
you can, okay. Well, we can discuss later. I'm not sure if if we are talking about the same thing, but if we are, maybe that's another way that I'm not aware of. Let's discuss. I'm interested because we are evolving this day by day. Basically, that's it. This was a bit of showcase. A cool thing. We interface with Plotly as well because every column is a variable. You can easily apply Plotly, so interactive plots, plotting the reduced dimension with the colors based on cell types or whatnot. And here I'm just showcasing that. For example, sometimes for complex data set is effective to plot the UMAP in 3 dimensions. You get some different perspective of of trajectories and heterogeneity. And so it's quite easy to do our single cell experiment. We calculate 3 dimension. Okay, didn't load some library. Maybe this one. No, maybe it's not. Yeah. Let me load this too. What did I miss?
Somewhere flatly.
Run new map. Where is run new map? Stranger. All the libraries should be here. Where is run new map? Who has a good memory? Skater.
About the get anywhere function?
Let me do it again.
Everyone should know about the get anywhere function. If you don't know where it is, but you think that some package may have loaded the package that it's in, get anywhere will find it.
Anywhere.
Get anywhere.
Get anywhere. Do the underscore now.
Yeah, GEC uppercase A. No underscore.
Anywhere.
And then give it the name of the function you're looking for. And if it's in any package that has been loaded by any package, you'll find it.
Okay.
Including the private functions.
I think the R needs to be lowercase.
Yeah, R is lowercase.
Now I'm tempted to implement a case instance version of get anywhere.
It's very cool, but anyway, I will show you the random version. So you basically calculate 3 UMAP dimension, same thing, same way you calculate 2. You plot these into the plot tree. You just declare the dimension as variable, the color as variable, so on and so forth. And as you can see, is with basically a couple of lines of code, you have a 3-dimensional representation where you can select cells and so on. I found these, useful in very few occasions, and it is quite good that we can do it quite easily. That's it. Now Michael will tell you how to do very fancy integration between the ecosystem.
Thank you. So I want to also make sure to leave enough time for questions. So I'm going to kind of jump through this, but again, you have the full rendered version as well. What is it here? So give a little bit of background. So I just learned about this tidy transcriptomic stuff and started using it last year. And before that, I had been playing out with think of a parallel set of packages, with the main one being plyranges. So plyranges was written by Stuart Lee, Dai Cook, and Michael Lawrence, who's around at the conference. And it's, again, a way that you can operate on genomic ranges using tidyverse. It's not changing the genomic range. It's performing what are called these endomorphic operations, meaning the same class that goes in is what comes out. And it's actually, I think it's even easier as far as an abstraction, because the G ranges is already tidy. It's one row per range, and it has metadata columns. So it's really straightforward, and it's very useful. Once you start using it, you realize this is actually quite a nicer way to work in some cases with G ranges. So then recently, we started talking about it's very nice to have all these different functions, and how can we bring them together. And so we've been seeing ways in which you can kind of combine the two. And so I have a little example here. So just to say what's this example going to do? Let's say we want to look at genes. So in our single cell experiment, we want to look at genes and group them by their location. Right? So up to now, we haven't been thinking about the locations of these genes. We have some that we're interested in by name from a paper, but let's say we want to group them by their location, maybe their distance to landmarks like ChIP-seq peaks. And so I found some ChIP-seq peaks for this is again just for demonstration. It's not a great scientific example, but like these are PBMC. These are cells from PBMC for full blood mononuclear. And I found some chip-seq peaks for the same cell type. It's not a good match, but just for the demo, okay? And so at the very beginning of this, I have to attach the gene locations. So you don't always have to attach gene locations. I have a note here, like if you use this package that I've worked on with some other folks in Bioconductor, you would already have the gene locations attached, but it's also very easy to attach gene locations using some transcript database. Maybe I'll switch back to the RStudio instance. So I'm just going to click through here. What I'm doing is I'm attaching gene ranges to the single cell experiment. Make sure I don't get any errors down here. Okay. Okay, so these are in HD38. And then I'm going to load this plyranges package. So plyranges, again, lets you operate on genomic ranges with familiar verbs. So slice shows you the first three. You can, select metadata columns using select. And so now I've gotten rid of some of the columns here using select function. And this is some operations to reduce down to the genes that are in the single cell experiment. So I'm selecting on the ones which are in the single cell experiment and sorting them, etc. And now I'm adding these ranges here, adding ranges to the single cell experiment. So it's nicer if you already have those there, but it's very easy to add them. Okay, so now I've got locations on the single cell experiment, and I can kind of start to combine the information from the single cell experiment and work with the ranges. This I'm jumping over. This is just code that was used to put the chip-seq peaks that I got from Annotation Hub, so it's a resource on Bioconductor, to move them into HD38. Not very interesting for today. So I just load them, but the code above is actually like what I used to bring them into HD38. So these are the peaks.
Exactly. I'm so happy that Bioconductor implements leftover without having to drop to the command line.
Yeah.
That kind
Oh, and by the way, one of our contributors here is building a new package that makes it even smoother. So you can check that out. It's his first package, so it's pretty cool. So I'm just gonna for the sake of time, I'm gonna take the top 5,000. Again, like not a very principled choice, but just to demonstrate this idea. And here's a more interesting operation. So I take the ranges now. So now we have genes of the single cell experiment located in a genome. Anchor 5P. This anchors the ranges at the 5' end. So for a gene, that's the transcription start site. Mutate width equals 1. This now takes from, instead of being a range of some extent, now it reduces the range to just the TSS. So now it's a single base pair range. And then add nearest distance, peaks, calculates the nearest distance from the TSS to these peaks that I have, chip-seq peaks. So it's very easy. Let me just step through that so you can see what the result is.
So this is wrapping a series of like G-ranges operations to annotate it to the nearest peak.
Yeah. So, and again, the key idea here is these are endomorphic, meaning same thing in and out, right? So that's ranges. This is apply ranges function, outcomes ranges. This is apply ranges function, outcomes ranges. Again, outcomes ranges with new metadata. Let me just run this line so you can see that.
So the anchor is a little bit like the group by where it applies to everything until you change the anchor.
Yeah, it's like in the background now, these are anchored ranges. They're still G ranges. You could still operate on them.
Is that like a subclass?
It's not a subclass. It's modifying the way in which, or I don't know, maybe I should ask Mike Lawrence if it's a subclass. It's just modifying the way that future apply ranges operations will work. So it's not modifying the way that they will work as geranges.
I have a question. Like when you apply the ranges to the genes, you got each transcript ranges, or you got the total gene ranges, or you got the main ranges and applied?
Yeah.
Like the second part of the question, like would you use this syntax to do transcript information, like do the same just for transcript information?
Yes. So you could do all of this at the transcript level, and it's like you planted question. I'm going to talk about exactly your first question in a lightning talk, hopefully today. So you can do, typically in bioconductor, if you ask for genes, it's the full extent of the gene, but you can do something better. And I'll just defer that maybe till later today.
We're gonna present it during the live.
Okay. I'll send you on an email.
All right.
Yeah. We have a new, better way to do that on in TXI Meta. But typically in bioconductor, a gene is from the leftmost to the rightmost position of all the isomers. Okay, so I'm going to hurry up because we just have a couple more minutes left. What I said I was interested in doing at the beginning, I have single cell experiment. I want to operate on the genes based on their position to some landmark in the genome. And so now I've got this distance. Let's see, let's look at that histogram one more time. So this is the distance of the genes measured in the single cell experiment, their distance to a ChIP-seq peak of the same, also from PBMC, right? So there's some that are actually, the TSS is in a ChIP-seq peak, maybe a promoter peak. There's others which are, between 0 to 10 kb, not that many. And then there's a bunch which are 10 kb to megabase, and then there's some which are even further away. So, you can see their distance to the landmark distinguishes these, and you can put these genes in different bins, right? So I just arbitrarily come up with these bins. So this is like 0 to 1 is they're in a ChIP-seq peak, 1 to 10 kb, or further all the way to, to the end of the chromosome, or there's no ChIP-seq peak on that chromosome. So I take this distance information and I basically put the genes into these bins. So just to show you what that is. And then at the end, so I'm in this process, I'm taking this information out of a ranges object and turning it into a table. And that's just going to help me like plot and left join later. So I have gene, distance, and now it's in a bin. And so we can, how many genes are in each category. So there's not that many in the middle. There's mostly actually in this far category. Let me make sure I didn't skip anything. Okay, so this is a nice new function in tidySingleCellExperiment, right? This ability to quickly pseudo bulk. So take the single cell experiment, pseudo bulk on a column, and then I add this distance information. It's a left join, so it adds it recognizes the gene column, it adds the distance bin. So basically every gene is in a bin, one of these 4 bins. And then I do what's called a nesting operation. And let me just show you what that looks like, and then I'll tell you what it's doing. So
just to clarify, the aggregate cells converts now a single cell experiment into summarized experiment. With the tidy view, you will keep seeing the similar representation, and keeps all the relevant metadata. You understand what you are grouping according to the result.
So this is, I think, very interesting. Now we have a table. On the left, we have the distance bin, and inside we have 4 summarized experiments. So these are we've chunked up the original summarized experiment, which represents pseudo bulk data. And we have these 4, which are now categorized by how far was the gene to a ChIP-seq peak. That's convenient because then we can operate on these without having a lot of control code, like for, this bin out of all the bins, all this like control syntax. You can move this all away and just focus on the operations you want to do. So for example, let's say I want to take the column mean. So that's, across all the genes for every pseudobulk sample. And I also want to preserve the cell type information. I can do that across the different summarized experiments. That's the genes grouped by their distance to a gypsy peak. I'm pulling this out as a table. So now I have the mean count for each cell type. And then this is just a smaller version of the cell type information. So last thing, I just show you the final baked cake is I grouped my genes by their distance. We pseudo bulked. We took the average of the counts across all the genes in the bin, and then we're showing that across the different cell types. So, there are many caveats along the way. This was mostly just for demonstration of what can you do? What are all the interesting things you can do? But what we see is the genes that are where the promoter is in a ChIP-seq peak, those have higher counts. And then we've just ordered these by the cell type. So I know that was, super fast. And if you read through I have some things okay, this step we could have thought about it differently, all these different caveats, but I just wanted to quickly show how to combine these two. Yeah, so we're at time. Maybe you have any last comments about where we're going or about the project, tidyomics project?
Yeah, I would reiterate. And
so you have seen that all this is very sexy and will save you a lot of hours of work. Is the same once you start. Is the same thing of tidyverse versus base R. I know many, well, having said that, you never lose any bioconductor tool, as we said. You can switch anytime for operations that are more convenient with some other grammar. So basically, you don't lose anything. It's just some other. We bridge to the tidyverse, which is huge for you to use, if you want to. The last thing I would say, I would just reiterate the invite. So this could be the easiest and most enjoyable authorship you can have, honestly. We want to make this paper involving the community, having all 5 continents and as many universities as possible. This is a joy for us. Actually, it's a good experiment that is currently working, but we invite even more people, as you can see. So this is, again, the QR code. I leave it 5 seconds there, and then I will show you the last, again, the project. As you can see, there are many tasks that are waiting for contributors. So we are many here. Some of the tasks are very trivial, so you don't need experience. Write a blog post, do a workshop in your institute, curate the documentation. There is a lot of documentation that can be improved with fresh eyes because we have been developing for many years. A lot of documentation and transparency can be improved. So you can give feedback and also work for accomplish that feedback. Last thing, that's the project, as I said. Very easy. You can just click, comment on the issue and propose yourself. We are here to help. We are doing a good thing. I think I'm very excited for this, and it's a beautiful thing for sure.
Yeah, I never mentioned the Slack channel, which I guess is the easiest way to talk to us and other people. It's on bioconductor tidiness in BioC with underscores. It actually predates both of us working and being interested in this, and it emerged at a bioconductor conference many years ago. Maybe 8 years ago or something. Yeah, so maybe any questions?
Yeah, so that's a very elegant tool, and I really appreciate your work. And so I have a question. So for all these pipe operators, are they multi-threaded? Cause I use a tidyverse well before, and I have a really big database, and it turns out that it takes a lot of time to wait on function. So I wonder if you do some kind of optimization for these pipe tools.
Well, two sides of this question. One is I want to reiterate that these are not analyses. So if you use parallelization on your analysis, this this doesn't involve that. But what this provides, that Michael showed very elegantly, that you can nest if you want to do some operation on every cell type or every sample, like normalization or whatnot. You can nest very elegantly the datasets, and there is FUR that use the map function. I mean, you need to know what PUR is and so on, but I'm just describing all the operation you do on the nested is very easy to parallelize, send to a cluster, with not much coding, actually changing one function for the other. So there is the future map that allow you to parallelize that. So in this case, we can parallelize analysis, given that you use the function that you use to analyze. But through the nesting, this can happen very elegantly and is all self-contained. So you're not creating global variables. So also that is quite robust.
Yeah, if I could have. So on the efficiency also, many things for exploratory data analysis, it's very fast. Like you won't experience any difference. We're not hiding anything. If you go to the open issues, many of them are like speed up this, speed up that. In plyranges, if there's many groups, like thousands of groups, certain operations can be slow. You'll see that on the open issues, and we're working on it. But I really, for many exploratory analysis, interactive analysis, plotting, all the things that Stefano showed, you're not going to experience a difference in speed.
Yeah, but all the optimization, as Michael said, are on top. So pick one place. If you are, a software engineer, those are the challenges that are for you.
Okay, great. We'll stay around if there's any questions. Thank you. Exciting to do it in person.